Every single day, your body performs about 1 trillion cell divisions. That's a million chances for something to go wrong. A misplaced letter in 3 billion letters of DNA code. In the mid-20th century, Frank Macfarlane Burnet and Lewis Thomas proposed a radical idea: that our immune systems weren't just for fighting off colds. They theorized a constant internal patrol, a concept they called immune surveillance. This wasn't about germs from the outside, but about policing errors from within, hunting for the first flicker of rebellion before it could become a threat. So how does this patrol work? Every one of your nucleated cells is constantly advertising what's happening inside. On their surface, they have proteins called the Major Histocompatibility Complex, or MHC Class I. You can think of them as tiny molecular billboards. These billboards don't display flashy ads. They display fragments of every protein being made inside the cell. A tiny piece of actin, a snippet of hemoglobin, a slice of keratin. It's a continuous, real-time status report broadcast to any immune cell that happens to pass by. But how does an immune cell, specifically a T cell, know which protein fragments are normal and which are dangerous? The training happens early, in an organ you might not think about much, the thymus. Here, developing T cells are shown all the normal protein fragments, the molecular signature of U. Any T cell that reacts too strongly to these self-peptides is ordered to self-destruct. It's a brutal but essential process of negative selection. Only the T cells that can ignore the healthy U are allowed to graduate and enter circulation. Now, imagine a single typo in the DNA of a skin cell. This mutation creates a slightly altered protein, one that has never existed in the body before. The cell's internal machinery, unaware of the error, chops up this new protein like any other and places a fragment of it onto its MHC billboard. This new fragment is a neoantigen. To a patrolling T cell, this isn't a status report anymore. It's a foreign flag, a signal that something inside this cell is fundamentally not self anymore. The T cell's receptor locks on to this MHC neoantigen complex. The fit is perfect, and the connection triggers an irreversible command. The T cell is a cytotoxic T lymphocyte, a professional killer. It presses up against the rogue cell and releases 2 types of proteins. First, perforin, which punches holes in the target cell's membrane. Then, granzymes, which enter through these new pores and initiate apoptosis, a tidy, programmed cell death. The potentially cancerous cell quietly dismantles itself from the inside out, neutralized before it can divide. This system is incredibly effective. So why does cancer still exist? Because cancer evolves. One of the first and most common tricks a burgeoning tumor learns is to simply stop advertising. It can downregulate its MHC Class I expression, effectively pulling its billboards from the surface. The T cells patrol, but there's nothing for them to read. The cell has gone dark. It still has foreign antigens inside, but with no way to display them, it becomes invisible to the T cell patrol. The immune system, however, has a countermeasure for this very trick. A different kind of immune cell, the natural killer, or NK cell, patrols with a different mission. NK cells don't look for foreign flags. They look for the billboards themselves. They operate on a missing self hypothesis. When an NK cell encounters a cell that isn't displaying the expected number of MHC molecules, it assumes something is wrong. This absence of a signal is, itself, the signal to attack. It's the bouncer checking for an ID, And if you don't have one, you're out. But the arms race continues. Some clever cancer cells learn to exploit the immune system's own safety mechanisms. T cells have receptors like PD-1, which act as brakes to prevent them from attacking healthy tissue too aggressively. Certain tumors can evolve to express the corresponding molecule, PD-L1. When the T cell's PD-1 connects with the tumor's PD-L1, it's like the tumor is pressing the T cell's off switch. This induces a state of exhaustion. The T cell is still there, it knows there's a threat, but it no longer has the energy or ability to attack. So when we ask why cancer happens, the answer is more complex than a single failure. For every tumor that is diagnosed, there are perhaps millions that were silently identified and eliminated. The cancers we see are not just random errors. They are the winners of a ruthless, microscopic game of natural selection played out over years within a single body. They are the descendants of cells that learn to hide, to deactivate their hunters, and to build defenses. They are the master escape artists, the ones that got away. Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.